For our next session, we have Dr. Carolyn Mathen. She is CEO and founder of biotech venture OCT Therapies and Research Private Limited. She is a stem cell biologist with 14-year experience in biotech industry. She holds a doctorate in zoology. She has earlier worked as head animal biotechnology with KITS Scientific Research Center, Mumbai. She has 4 patents and several papers published in national and international journals. She is the winner of prestigious big grant under BIRAC DBT for the proposal titled Establishment of Stem Cell Bank as an Alternative Skin Bank. I welcome you, Dr. Caroline, for the talk.
Thank you, Renuka, for that introduction. And it's my pleasure to be here. Thank you for the invitation to come and share about this latest cutting-edge technology that is stem cells in cosmetology. So I am the co-founder of a company called OCT Therapies and Research Private Limited. We are a biotech company and we are focused on developing both cosmetic and clinical applications for stem cells. And our current focus is on developing very cutting-edge stem cell therapy for wound healing in both burns as well as in non-healing ulcers. This is our vision. We want to be globally recognized as one of the leading biotech companies and, like I said, focused on stem cell-based therapies to meet critical unmet needs. And our mission in brief means that we are, in it for the long run, no shortcuts to reach where we want to be. We want to be the best in our field. And we are going to do that by being an innovation-led company, adhering to the highest levels of quality and safety in whatever products we develop. So there is a lot of hype about stem cells. You must have heard there is a hype, there is a hope. So how do we distinguish between, the chaff from the grain, so to speak? For the uninitiated, I'm going to give you a brief introduction about what are stem cells and why it is so exciting for us today. So, by definition, a stem cell is an undifferentiated cell of a multicellular organism. This means that if you think of, our own bodies, like we have different tissues in the body, you have your heart, liver, skin, and so on and so forth. So these are specialized cells or differentiated cells with specific functions. These cells are not going to divide and form something else. A stem cell is something which is undifferentiated. That means it has the potential to form any cell in the body depending on what cues or signals you give it. So now these cells are special. So if you think of a normal cell which divides, you will have 2 daughter cells which are similar to the parent cell. But in stem cells, it's different. It's called an unequal division because it has 2 kinds of daughter cells. One daughter cell will resemble the parent cell and the other daughter cell will be a specialized cell with a specific function. So if you look at classification of stem cells based on potency, we have totipotent, pluripotent and multipotent. So toti means total potency. So you have to think of an embryo along with the extra embryonic membranes which can actually form a full organism. That is totipotent. Pluripotent will be like these embryonic stem cells which can form the 3 different lineages. That is, you have the ectoderm, endoderm, mesoderm. So that is pluripotent. And multipotent, these are the adult stem cells. They have limited potency compared to the pluripotent and the totipotent cells. So say for example, if you look at a nerve cell, so a progenitor cell is nothing but the parent cell and these cells can form different cells, that is neuron, astrocyte, oligodendrocyte, but these are all belonging to the nervous system. So similarly, if you think of a hematopoietic stem cell, a hematopoietic stem cell is nothing but which belongs to the blood lineage. So that will form only different cells but belonging to the blood lineage. So it's a single lineage differentiation. So I've already spoken about embryonic stem cells and adult stem cells. Now there is a very new kind of stem cell called as the induced pluripotent stem cell or iPSCs. Now the first 2, that is embryonic and adult stem cells, they are occurring naturally in nature. So they occur in our different body tissues of the body. By the way, I just want to mention that I'll be concentrating on animal stem cells and not really on plant stem cells because I'm not a botanist. Anyway, so the other cell that I was talking about, these iPSCs, they are produced in the lab. So how it is different is you take a differentiated cell. So say for example, you can take a skin tissue. So from the skin tissue, you have keratinocytes or you have dermal fibroblasts. So these are already differentiated. You de-differentiate it by doing genetic engineering so that it goes back to the embryonic stage. And then you can do a direct